go. Right, so this metagenomic high-C project is something that has been going on a little bit at UC Davis, and also we've had a group at the University of Technology in Sydney. Matt DeMayer, who's in the room here, is currently doing some analysis on this. So I'll give you a bit of background on how we arrived at this approach. And it really starts a long time ago, 10 years ago, when I was in the labs of Fred Blattner and Nicole Perna working on the genomes of the first few E. coli. And when we finished sequencing these first few E. coli, the very first thing that we did was to predict the genes present in the genomes. And when we did that, we found this kind of shocking pattern where we saw that of all the genes predicted across all of the genomes, is this the laser pointer? Okay. Here we go. Only about 40% of the total gene content across these 3 strains of E. coli was common to all of the E. coli. So each one of these strains, even though they're the same species, is actually remarkably different in terms of their gene content. And the other thing that surprised us a bit was that each one of the strains had a relatively large complement of unique genes. Now, these 3 strains in the human host are actually behaving quite differently to each other. So one of them is a commensal, the MG1655 strain. Another one is uropathogen. And this one, you really don't want to get it. It causes nasty, bloody diarrhea. We had a few incidents with that in Wisconsin. Now, if you think about what people are doing today when they're doing microbial community profiling, people are going in with 16S ribosomal RNA amplicon sequencing. And these 3 organisms are members of the same species. They have almost identical 16S ribosomal RNA sequences. And yet, if you found an E. coli 16S sequence in the human host, you basically have no information about which of these 3 you have in that person. But it really makes a pretty big difference to that person if they've got, something that might cause nasty, bloody diarrhea.
Did you see that paper that Rob Knight and those guys just published saying all they need is the 16S? It's in this mid-journal like this week.
Oh, really? Okay, I'll have to go read that one.
It's complete crap.
I don't know if I believe that. So fast forward a few years for me, when I was in the lab of Jonathan Eisen, they published this paper about the Genomic Encyclopedia of Bacteria and Archaea. And in this project, what they sought to do was to pick the collection of organisms which were in culture and had the maximally diverse 16S ribosomal RNA sequences, and they selected those for genome sequencing. And what they were able to show is that when you pick organisms in this way, you discover new gene families at the highest possible rate. So at a much higher rate than if you limited yourself to sequencing a single species over and over again, or a family or a phylum even. And so there is this basically strong correlation between novelty in the 16S ribosomal RNA and novelty in the gene content of organisms. And so meanwhile, of course, we've discovered that there is an enormous amount of microbial diversity on the planet. This is a figure showing the amount of phylogenetic diversity represented in our 16S ribosomal RNA databases. And the dark green line here is showing you the amount of diversity in things that we know how to culture and that we have names for. These are things that might be in the taxonomy. And the light green line here is showing you the amount of diversity represented in things that we've never cultured, and we don't necessarily have names for them. And so, 10 years ago, it was kind of like living in a small town. We had names for everybody, and we knew who was there, and we could grow them. But now it's clear that we're actually living in a gigantic city, and we don't know who's around us. So metagenomics has been proposed as a mechanism which would allow us to capture the gene content of uncultured microbial communities and basically reconstruct their function. But metagenomics, the way it's currently practiced, is absolutely terrible. I hate it. And it has really huge problems. And those problems come in really in 2 steps of the classic metagenomics pipeline. So what you typically do with metagenomics is to take a sample, and the very first thing you do is smash up the sample into little pieces so that you can get the DNA out of the cells. But when you do this, you're creating a big problem because you've taken DNA that was nicely compartmentalized in each one of the cells in the sample and kind of mixed it all together. So you no longer know which bit of DNA came from which cell. So then you go on to purify the DNA, and then you shear it again so that you can get fragments small enough to load into your favorite sequencer. And the problem with this shearing process is that you've taken the information contained in nice long strands of DNA about which genes were together on the same molecule and destroyed that information so that you can load your sample into a sequencer. And then what ends up happening inevitably is that a biologist will rock up to someone like us and ask us to undo the damage that they did in these two steps computationally. And that's really hard. I mean, people have been working on binning and metagenomic analysis for the last 15 years, and we've made progress, but we're still a really long way from being able to reconstruct the individual genomes that were present in these samples. So how are we going to solve this problem? Well, I can think of a few possible ways. One would be to physically dissect the microbial communities. And this is something that I've participated in a little bit in a project to do single-cell genome sequencing. That has a lot of potential, but one of the things that we showed in that project was that if you wanted to characterize just half of the known diversity on the planet now, you'd have to go out and find and flow sort and single-cell sequence 20,000 cells. And doing 20,000 of them maybe is not so bad, but you actually have to find the right 20,000 cells to describe the diversity out there on the planet. So that's probably going to be too hard for us. So another possibility might be to come up with better inference methods. Maybe the people who have worked on binning so far just haven't been smart enough, and there's some clever way that we could solve this problem. Well, I'll talk a little bit about that. And then I'll talk about these other two possibilities later. So I thought kind of naively and a bit arrogantly that I may be able to improve the state of the art in computational analysis of metagenomes. And in doing so, developed this piece of software called PhyloSift. With PhyloSift, what we're doing is we're basically taking input sequences, raw metagenomic reads or 6' amplicon reads, and aligning them against a reference database that contains 37 elite gene families. This is an updated set of gene families to what you guys had been using in in Phylopithia Plus.
Oh, can we use those?
Yeah, of course, they're publicly available. So these include bacteria and archaea, whereas the previous had only covered just the bacteria.
What the hell does this mean? Sorry.
So these are single copy genes that are present in almost every known bacteria and archaean. And they appear to have relatively congruent phylogenies. With each other. So they all appear to have the same phylogeny, if you infer phylogenies on each gene separately.
For a suitably chosen set of fairly distantly related ones.
That's right, yeah. Yeah, so the pipeline searches the reads against those families, and it picks just a few representatives that we don't have to search against every genome ever. We just have to search against a small handful of representatives. And then it adds any reads that are found to be homologous to a gene family to an existing multiple sequence alignment using HMM align. And that's nice because then you can get a large multiple alignment containing all of the input sequences that were homologous to the family in essentially linear time. And then you take that big multiple alignment and you use it to do phylogenetic placement against a reference tree, which represents each of those gene families. And so we are using pPlacer for this, and that's actually quite a nice algorithm because it can do Bayesian phylogenetic placement. And what that allows you to do is that for a particular sequence, and this is especially important for short read sequences, it allows you to calculate analytically the posterior probability that sequence evolved from each one of the lineages in the phylogeny. So you get a probability distribution over the phylogeny for every sequence. And from that, we can then do various types of analyses, like multi-sample comparisons using principal components analysis, or various types of visualization, or Bayes factor tests for the presence of a particular organism. So as I was developing this software, the human microbiome project was really ramping up. And my partner at the time became pregnant. And we got it in our heads that really there was, as scientists, no greater gift that we could give to our expecting child than a catalog of his developing gut microbiome. And so on December 3rd, 2010, my son Kai was born, and his microbiome was born. If you've never seen an infant microbiome, they're kind of nasty, sticky things. But we loved him and his microbiome. And from that point on, we went to go on parental leave in Sydney, Australia. And thankfully, he continued pooping. As you can see, there was a lot of poop here. Um, and over the course of several months, we continued to collect his bowel movements off of the nappies. And about 2 months in, we actually had a bit of a crisis in the family because my partner was diagnosed with a bacterial infection. And she was prescribed antibiotics by the GP. And we were faced with this decision then of whether we continue to breastfeed our child or do we put him on formula? Because we read up about these particular antibiotics and realized that they were going to passage through into the breast milk unmetabolized. And so if we continued to breastfeed, we would in effect be giving our child antibiotics. And so we didn't know much at the time about the effect that that would have on an infant, but our instinct was to take him off of breast milk and put him on formula for that week. You can see that shown here. There's a week when he was on formula. And so then at the end of our parental leave, we were faced with this little problem of having a lot of poop in tubes and no idea how we were going to get it through the quarantine in the United States. But luckily, Mark Brown and Tiffany Nelson came to our rescue. And so these guys are kind of the unsung heroes of my quest to sequence baby poo because they extracted the DNA and mailed it into the United States for me. So this project then became a little bit of a hobby of mine, but it was something that I didn't exactly have any funding for. And so I decided to come up with clever ways of trying to get sequence data for these samples. And I got lucky again because a guy down the hall from me had recently worked out that he could basically do a homebrew version of the Illumina Nextera library protocol. And so the way this works is that you take the TN5 transposase that's used in the Illumina Nextera kit and purify it off a plasmid from an expression host. And then you can charge that with custom synthesized oligos that contain barcodes. And so this allowed us to generate a large number of Illumina libraries with very little money. And so we went on and got sequence data from that. And of course, because I was working on PhyloSift, I wanted to take the sequence data and put it into PhyloSift. And when you do that with one of these samples, you get a picture that looks kind of like this. So this is the reference phylogeny for the whole tree of bacterial and archaea life. And branches that have been colored red here are branches where you have some amount of probability mass for a reed deriving from that branch. And when a branch has been thickened, like this one, it's saying that you've got a lot of probability mass, or many reeds deriving from that branch. And so you can see this really thick branch right here leads to something called Bifidobacterium longum, subspecies infantis. And that's, of course, exactly what you would expect to see in the gut of a breastfed infant, because that's one of the bacteria that's known to digest human milk oligosaccharides. So what happened when he was on formula? Well, we went ahead and we analyzed all 45 of the samples that we had. And the first analysis we did was to calculate phylogenetic diversity on these placement trees. So if you plot that for each of the samples with respect to time here, you can see that initially it was somewhat high, and then there was a shift downward, which I'll come back to. But when he was on formula, those are the points shown in red here. Initially, there was a big spike in phylogenetic diversity, and then a crash. But immediately following the resumption of breast milk feeding, the diversity went right back to where it was before. And we were, of course, overjoyed to see this as parents because we had no idea what was going to happen to our little guy, although he definitely seemed happier when he got back on breast milk, I'll tell you that much. Another way of looking at it is using something called balance-weighted phylogenetic diversity. You see a different pattern there, but without going into the details of the methods, I'm not going to say much about the differences. So another way of looking at these microbial communities is to do something called phylogenetic edge PCA. So whereas the previous analysis was talking about alpha diversity, diversity within a sample, this is looking at beta diversity, a comparison of the community structures, basically. And there's some nice mathematical properties of phylogenetic edge PCA, but I'm not going to talk about those. What I will tell you about is that we're able to describe a large amount of the variation among the samples using this method. There's this interesting pattern with respect to his age. So early in life, there was a lot of variability in the community structure in terms of the abundances of the different organisms. They were changing every day. But about a month and a half in, it seemed like the community settled into a stable structure, and we saw the exact same microbial community every single day. It was dominated by Bacteroides. And so you can see that on this first principal component. This component corresponds to an increase in a specific lineage of Bacteroides. And so the samples over here, which correspond to around 2 months in life, were dominated by that organism, which is commonly found in the adult gut. And so then what happened when he went on to formula? The biggest effect on him, actually, it made him really unhappy. He got stopped up, and he didn't have a bowel movement for 3 days. And then on the 3rd day, he had 6 bowel movements in 1 day. He just got it all out of him. And he felt a lot better than that, I could tell. But what happened over the course of the day was that the microbial community seemed a little bit strange. So previously it had been over here. So it was a little bit different. And then it kind of went all over the place over the course of the day. But by the evening, it was right back near that canonical community structure. And so we've got an N of 1 here, but the way we're kind of speculatively interpreting this is that the formula didn't actually change the native gut community. What caused these shifts in community structure was the amount of time that the material was in his body. So my partner is a population geneticist, and she does statistical genetics. And so we got a little bit interested in looking at the population genomics of the dominant organism, Bacteroides feces here. And so the way we did this was to take the reads and map them back to the Bacteroides feces reference genome. And when we do that, you can see the total number of reads, or the fraction of each sample, that maps back, shown against time here, and that increases over time. So this sudden jump in the abundance of this organism corresponds to that shift to a sort of stable community structure. What we did once we had the reads mapped was to identify variants relative to this reference genome using the low-freq software, and we called single nucleotide variants at each time point. And we found a large number of variants. When we plot the abundance of the variants at each time point, we saw this kind of confusing pattern where each variant was either almost completely dominant at 100% abundance, or they were at really low abundance across the time point. So the question then becomes, how are these variants linked to each other? So if you plot then the variants that are present in each sample relative to time, this isn't really showing up very well. Some projectors it works well on, but anyway, we saw this pattern where large numbers of variants were coming and going together. And so that suggests then that we may have 2 strains here. However, there were a lot of variants that kind of broke from this pattern a bit, and so there may actually be some recombination, which we did a bit of analysis on. But the major questions that come out of this is, how do we evaluate whether a handful of single nucleotide variants are actually occurring in the same cell? And how tightly linked are they around the genome? Short-read data doesn't give you this information, because with short-read sequencing, you're only able to link variants across a few hundred base pairs. And a related question then, of course, is how many strains are actually present in one of these systems, and how are they recombining with each other? So this brings us back to metagenomics, and how we can basically get more linkage information out of our metagenome data. Nanopore long-read sequencing might be one way of doing this. I think there's going to be a lot of potential there, but there are questions that will never be answered with nanopore long-read sequencing. So I'm now going to tell you about something a little bit magical, I think, which is called Hi-C. So Hi-C is a technology that was originally developed to capture the 3D structure of chromosomes. And people applied this in cancer genomics recently to basically reconstruct the entire spatial architecture of cancer chromosomes. And the way this works is to basically take your sample initially in its native state and cross-link it using formaldehyde. And what that does is it kind of glues together proteins and DNA that are bound to each other. And you can get structures like this, where you've got 2 different strands of DNA. These might be like plasmids and a chromosome that are both bound by a protein. And they become cross-linked to each other. So then you go ahead and you lyse the cells, and you cut them with a restriction enzyme, and you fill in the ends, the overhangs there, with a label like biotin, which allows you to pull those down later. And before you pull them down, you dilute the sample and ligate it under conditions that favors these so-called proximity ligations, where strands of DNA that are co-bound by proteins will ligate to each other. So you can pull these down and then sequence them using Illumina technology. And what you get out of this are a collection of reads that basically tell you which bits of DNA were physically nearby each other in the sample that you cross-linked. So people have been applying this to reconstruct 3D chromosome architectures. We actually thought and speculated that this might be useful for metagenomics, because basically bits of DNA that are together in the same cell should become cross-linked to each other at much higher rates than bits of DNA present in different cells. And so we set out to do a little experiment to test this, where we took 5 organisms which had really well-defined reference genomes, and we cultured them up in the lab and mixed them in approximately equal parts based on their optical density, and applied the standard Hi-C protocol to make a Hi-C library from that mixture and sequence it on the aluminum IC. And in order to basically test how well this worked, the question we wanted to ask was whether, using that Hi-C data, you could take contigs from a metagenomic draft assembly and link the contigs that came from the same organism back together into a single cluster. So if you take Hi-C data and map it to a metagenomic assembly and then draw it as a graph, you get a picture that looks like this, where the nodes are contigs, and the size of the nodes is proportional to the length of the contig. And links between nodes are representing Hi-C read pairs that have linked those 2 contigs to each other. And so if you just calculate an automated layout on one of these graphs using the Fruchterman-Reingold algorithm, you can already see that we're starting to get some structure in the graph here. So does that structure line up with the species that we put into the mixture? Well, if you colored the nodes by the species, it turns out that yes, in fact, this clustering of nodes does line up quite well with the species. So this is E. coli. There's actually 2 strains there. That's why you see so many nodes, because of strain-specific content, creating more contigs in the assembly. But we then wanted to figure out whether we could actually automatically calculate these species clusters. And it turns out that after fiddling with the parameters a little bit, you can use Markov clustering with the right inflation parameter. And you'll get 4 clusters, each of which correspond essentially to the entire gene content of the species that we put into this sample. So, of course, then we wanted to know why this seems to be working so well. And so we sought to characterize then what the specific signal in the Hi-C data was that was allowing us to do this species clustering. And so the first thing that we discovered is that 99% of the Hi-C read pairs are associating gene content from the same cell. So there's a pretty high signal-to-noise ratio there. The next thing that we looked at was the insert size, basically how far apart read pairs and Hi-C data were mapping. And what we found is that basically the Hi-C read pairs span the entire chromosome. So you can get read pairs that are linking 2 genes on the opposite ends of a circular chromosome. And those are relatively common. I mean, there is this kind of exponential decay in the insert size. So you're getting more reads that are linking things nearby each other on the chromosome, but you're still getting reads that are linking things very far away. The other really interesting property relates to what Hi-C is doing with plasmids. So we put this lactobacillus into the mixture, and that contains 2 relatively small plasmids along with the host chromosome. So we found that the Hi-C data was linking the host chromosome back to the plasmids at several orders of magnitude higher rates than it was linking material across cells. So that suggests that you can actually associate mobile elements like plasmids back to the host chromosome using Hi-C data. So how would you use this then to try to resolve strain differences? Well, we wanted to characterize whether this type of information or data was useful for resolving strains. The way we did this was to define a graph where the nodes were single nucleotide variants in the E. coli genomes, the 2 strains of E. coli that we put into the sample. And if you then draw a graph where you link up the nodes whenever you observe them in a read pair, and then compare the graphs that you get for mate pair types of data, 5, 10, 20 kilobase mate pair reads versus Hi-C reads, you see that in the largest connected component in this graph, you're only able to capture up to about 30% of the nodes in the graph using these large insert mate pairs, whereas with the Hi-C data, we're able to capture nearly the entire graph. And so if you want to do haplotype resolution, you actually really need to be able to have all of the graph in a single connected component. That helps a lot. So intuitively, you can think of this as the Hi-C read data making both local and global connections, whereas the mate pairs are only making local connections within the insert size that the library was prepped to. So a related property of this is the number of hops that you would have to take in that graph to transit from basically 2 randomly chosen endpoints. And so in mate pair data, if you pick two nodes at random in that graph and basically calculate the path length between those two nodes, that path length grows approximately linearly with their distance on the chromosome, as you would expect. But in the high-C data, it turns out that that path length through the variant graph is essentially unrelated to the distance that the variants are in the chromosome. And so in the E. coli, for example, you can walk between any two variant sites in the E. coli chromosome in, on average, 4 or 5 hops. And so that's nice because if you're trying to then resolve haplotypes, you can imagine that your probability of linkage and that error in that estimation is going to compound with each one of these hops that you take.
Sorry, I mean, this technology was built exactly to see where you don't get this link, where you get the disequilibrium and the linkages, right? So like Kleckner and all those guys, when they're trying to do, these are the domains that are sticking out. And so how come you get that in coli? I mean, that's coli and salmonella is where they did it. So why are you not picking that up? Does that make sense?
We do get the domain structure, yeah.
You do see the domain structure?
Yeah, the domain structure is there. It's just that it seems like there's enough variability from one cell to the next in terms of which bits are associating that you actually still are able to transit from one bit of the genome to another. Relatively small number of hops. Yeah, so we, it turns out, weren't the only group thinking about this. And a couple months after we published our paper online, another group came along, Jay Schinder's group, and basically reproduced very similar results to ours. They didn't do so much analysis of the strain resolution, but they did show that they can do this really nice species clustering on a different sample that was created in the lab. So you don't have to take my word that this works. So one question might be, how do we actually use this long-range linkage information to resolve strains? Well, if you imagine taking an assembly graph, like a string graph, for example, where the nodes correspond to a sequence and the edges basically show the adjacencies in sequence around the genome, you could overlay high-C information onto that, where you add these weighted edges, which indicate the probability that particular variant regions, so for example, strain-specific variants, are actually linked to each other. And then you can apply any number of algorithms to basically cluster those variants with each other. One possibility might just be to enumerate all paths and then calculate their likelihoods. So I think I should probably emphasize that this is really just the beginning of what we're going to do with high-C. There are a number of open questions here. We've demonstrated that this can work on a very carefully controlled system with cultured isolates, but nobody, to my knowledge yet, has actually applied this to environmental systems. So there's a lot of opportunity here. There's also a lot of work to be done with the computation. There's a lot of effort that would need to go into resolving the strain genotypes. There's also a lot of scope for improvement of the lab protocols. It's finicky and slow and expensive. Most of the protocols use 6 cutters because they're focused on human genomes, but 6 cutters only give you information every 10 kilobases or so. And that's a pretty big chunk of a microbial genome, so you might prefer to use a 4 cutter there. But I think there's still a lot of value in thinking about this because there are potentially a number of really interesting applications that we could target using this kind of data. So a few questions are whether frequently interacting microbes end up being associated using high-C data just because they're physically next to each other in a sample. Can we link viruses back to their hosts? We showed that we can link plasmids back to a host chromosome, but viruses you would expect to be invading cells, and so potentially you could get some of that information. And can we look at predator and prey dynamics? And I'll refer you to Chris Beitel's blog if you want to get some more ideas. He's one of the students who's been leading aspects of this project. With that, I'll end. I've got a lot of people who have contributed to this work, some of whom are here today. And of course, a lot of the lab work on this initial bit was done at UC Davis. But we're now at UTS and working hard on trying to improve this approach. So thanks.
I didn't really get how you actually cross-link it. So this protein, so this structure, the chromosome structure is already in place and you just have to cross-link it, or do you have to add some sort of protein?
Yeah, we don't add anything except 1% formaldehyde. So the standard cross-linking protocols for Hi-C take the sample and drop it in formaldehyde for 30 minutes.
Okay.
So you just use the naturally occurring proteins.
How much more expensive is it than currently?
It probably, yeah, that's a great question. I don't actually know of anybody offering Hi-C as a service. If that gives you some idea.
Money cannot buy.
I think I saw a slide flash by that it's not directly, you're not able to directly sequence it or something. There was a note, something about not sequencing and simulating or something.
Oh, right.
I didn't catch it.
That was probably on the assembly slide. So if you just do a Hi-C library, the read pairs that you'll get back contain, at times, they contain unmarked ligation junctions because you've done that ligation. And assemblers really don't like that. So you have to separately generate a normal metagenomic shotgun library to do your reference assembly that you can then link with Hi-C.
I guess maybe a follow-up quick question was, so besides cost, are there any other apparent sort of scaling issues that you think you might run into at this point when applying it to big metagenomic data sets?
Oh yeah, I mean, there's tons of them. I mean, with normal sequencing libraries, we've gotten really good at barcoding them. And so, you can make 100 libraries and sequence it in a lane. But people haven't come up with, schemes to introduce the barcodes early enough in the Hi-C library prep process that you can then, basically pool things and work on one sample. So the amount of work per sample is quite high for Hi-C in the lab. And then, of course, the analysis tools aren't really there. So we need people working on computational methods to analyze the data.
But why do you need to barcode it?
It would just be an efficiency question in the lab. If you can, barcode a whole lot of samples up front and put them in one tube and then just handle a single tube, it saves people in the lab a lot of time. It saves on
It's a lot
reagents.
cheaper. Can you go back to your graph of
Sure.
the relative occurrence of the chunks there?
Oh, so I should point out that color scale is log.
So are those jumps?
Yeah,
Are those real?
these jumps, like the
No,
just shifting
the,
color.
The actual graph where you've got the insert distance.
Oh, that. Yeah, they're not real. Well, they might be real. I don't know.
If there are particular domains that are interacting with each other.
Yeah, I wonder if that's your domain structure.
It might be part of it. I mean, there's also a lot of restriction site bias too, right? Because you only get data next to the 6-cutter sites
You're using
from the
6
HIN3.
cutters only.
Yeah.
So you don't get the whole. If you have two sequences that differ in 6 SNPs, you will never get that out unless they're next to your inserts. Is that right? Sorry, I'm just trying to understand.
Yeah, I mean, you need the site to be within range of a 6-cutter. So what you actually need is because you get information both from the parent library, which gives you some amount of local information about the linkage of SNPs,
Okay.
along with the Hi-C library, which gives you a bit more global information.
You could use that, like the Sage technology. I use those type 2 restricted enzymes that reach out and cut at just a distance. That would actually get you around that. So you wouldn't need to worry about how many restriction sites you have.
Yeah, that might be. I mean, we were thinking initially we might just try to switch to 4-cutters.
But 4 cutters are also biased.
Yeah. Okay. Thanks.